All right, so we're back talking about the process of science in part 3 here, and we left off with how to run an experiment, the steps behind the scientific method to be able to say this idea is backed by science. As I mentioned in the previous lecture, you want to go through these steps to try to increase and ensure as much accuracy as possible. And if you think it's accurate, you think your conclusions are solid, as I mentioned before, send it out for peer review. We don't want to trust something that has not been tested rigorously and peer reviewed and has had a large sample size. Those are all very, very important elements when it comes to trusting science versus pseudoscience. You know, the carnival lady with the crystal ball, does she go through this process? That's the thing. That's the reason why we want to look at science differently. So, all right, so let's talk about how to set up the experiment. That's going to be important. And I can't stress enough, when you're designing an experiment, take meticulous notes. How big was this? How hot was that? How many grams of that? All that information is very, very important when we're designing that experiment. Because when we get that data and we get those results and we start to draw conclusions, we want to make sure we have a very, very precise process, because then we're going to hand that process off to somebody else and say, here, you do it and tell me if you get the same results. It's like whoever created the very first recipe for beer or baking a cake or making whatever, they went through this process and then they crafted the recipe and they said, here, somebody else, see if you get the same results. This is how our recipes are produced, through this process. So, okay, so let's take a look at how do we go about designing a good, credible experiment. So we start with a hypothesis. Your hypothesis is your tentative explanation, kind of a guess, I think. But the thing to make sure when you write a hypothesis, you want to write it so it can be supported or rejected by the data. So here's an example of a bad hypothesis. All right. I think some bacteria will die if exposed to bleach. How many are some? How can we get results? So let's say we're going to put bleach on a dish of bacteria and see if they die. How do we know they died from the bleach and not from something else? What's measurable? Some is not a measurable number. So a better hypothesis is giving exact numbers. I predict that 30% or greater of the bacteria will die when exposed to bleach. Now I have a number. Now I have a tangible number that I can measure to. So you run your experiment, you put bleach on your bacteria, and let's say 28% of them die. Do your results support your hypothesis? No, they don't. But this is where we get into that redesign. Do we redesign the hypothesis and tweak that number? I say maybe you're going to go to 25% or whatever. But just make sure when you're writing a hypothesis, it's something that can be supported or rejected by the data. And again, it's a tentative explanation about what we think should happen. So we got a good hypothesis written, and we're going to design our experiment. We need to figure out What are we manipulating? This is known as your independent variable. The independent variable is what is being manipulated. What are you changing? Manipulate sounds like or has a negative context to it. So what is changing with the experiment? Is it the amount of bleach, the amount of time, the temperatures? Those are all independent variables, things that are being changed within the experimental design. Now, the dependent variable is what is being measured. So you're measuring something. You're measuring the reaction rate. You're measuring the amount of growth. You're measuring the whatever based on how much time you let that plant grow or whatever the case is here. This is what you're collecting your data about. You're measuring it. You're measuring in grams, in liters, in volume, in time, in whatever. These are usually the things that get measured, are the dependent variables. Now, within the experiment, you need to have a standardized variable. The standardized variable is what is being held constant. So you're not changing any of this fact. So if we're talking about plant growth, how much will a plant grow given 4 hours of sunlight, 8 hours of sunlight, 10 hours of sunlight? So the dependent variable is the growth of the plant. The independent is the amount of sunlight. The standardized variable, you better use the exact same type of plant. All tomato plants. All the same type of tomato plant. You don't want a red and a yellow and a orange tomato plant, the plants that produce orange tomatoes. You want all the same type. Maybe 12 Roma tomato plants are going to be used for this experiment. Now, something to think about is sample size. Oh. Sorry, got to get my typing better. So, how large of a sample will yield results we can trust? And this varies. Some experiments you might need thousands of samples. Others you might need dozens of samples. But what I can tell you with science, the larger the sample size, the more validity we typically see. Flip a coin 3 times. Flip a coin 300 times. Which one's going to give you more accurate results? You might get lucky with 3 flips and get all heads. Does that mean that there's no tail? Not really. But a bigger sample size is better. Okay? Bottom line, bigger sample size is always better. All right, so now we got our experiment set up. Maybe we're doing the plants, we're doing the bacteria, we're doing whatever experiment. We need a control group. All right, so a controlled group is a group that is not exposed to the independent variable. So if we're talking about that plant growth thing, I'm not going to expose them to different variations of sunlight. They're going to all have a standard amount of sunlight. Pick a number, 10 hours. Just the number that popped in my head right now. My control group all gets 10 hours of sunlight. The other plants get 4 hours, 6 hours, 8 hours, 12 hours, 14 hours. Get the point? Get the idea? Control group's your standard. This is what you're comparing against. Now, the control group often gets a placebo, depending upon the type of experiment. So the placebo is a fake substance to make you think you're in the control group. So if we're back to that earlier experiment we talked about in the previous part, excuse me, when we talked about the vaccination, you got to give every kid a shot. You can't give 50 kids a shot with the vaccine and 50 kids no shots, because then you know for sure who is not getting the vaccine. So 50 kids get a shot of saline solution, nothing in it, salt water, but they don't know they are getting the placebo. You don't want to tell the control group they are the control group, because that can impact and affect how they behave, and that can skew results, things like that. Now, sometimes placebo effect happens. And this is when people get this, oh, it's working, it feels good. It's like giving somebody a non-alcoholic drink and they think they're drunk. That's a placebo effect. Now, there is some science here. There are neurotransmitters in the brain that get triggered sometimes when people think they are part of this study, but they're actually getting a placebo. And again, they don't know they're getting a placebo. Okay, I got my shot. Now, they don't know if it's the vaccine or not, but oh, that's helping me. That's really making me feel better. That's protecting me. And what they're doing is triggering neurotransmitters in the brain to be produced that then can make you feel better. So is your feeling good from the actual independent variable? No. But does it matter if the placebo effect works? If the placebo effect makes you feel good, sure, that's great. Now, in the case of a vaccine, placebo effect is not going to give you protection at all against a virus. So sometimes placebo effect can be used, and great. Other times, it's not doing any good. We don't want to rely on a placebo effect to keep us healthy in certain situations. Okay, so that placebo effect is what can trigger what we call pseudoscience. Oh, let me get the font bigger, sorry. Didn't realize it was that small. Let's go like this. Boom. Okay, so the placebo effect can trigger pseudoscience, or pseudoscience is triggered by the placebo effect. Like, hey, I'm feeling really good because I did this. All right, so now then people are trying to promote this as scientific, and they're relying on the placebo effect. The challenge is placebo effect doesn't always work for everybody. You get a lot of variation in there. So when we're running these research projects, we often do a double-blind study. The researcher often doesn't know who is in the control group versus the test group, because you don't want the researcher unintentionally skewing the collection of the data, skewing the results as they collected the data. So if we're vaccinating 100 kids, I'm running this research project, somebody else is sticking the needles in those kids, and somebody else is tracking who has the vaccine versus who doesn't. I do not know that when I then go to collect the data, because I don't want to bias the data. All right, so lots of things we need to do to have a factual, rigorous scientific study. This is why when people say, I did my research, did they do any of this? Or did they just Google a bunch of stuff and read some articles and get an idea from it? All right, we'll pick up more in part 4.